Welcome to the SANS Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump into today's deep dive, just a quick reminder to hit that like and subscribe button, and please share this with your friends and family if you find it interesting. It really supports the SANS Science Experience. Okay, so today we're focusing on something pretty groundbreaking. It actually changes how we think about, the fundamental drivers of cancer. We usually talk about genetic mistakes in the main nuclear genome, right?
That's the cell's main instruction manual.
Exactly. But today, we're heading into the mitochondria, the cell's little power plants, because researchers have found a completely new class of, really aggressive cancer mutation there in the mitochondrial DNA, the mtDNA.
Yeah, this is huge finding. And it's all about these very specific recurring mutations hotspots that they found in the mitochondrial ribosomal RNA genes, the mtRNA genes.
Ribosomal RNA in the mitochondria.
And the implications are pretty profound because it's not just like a minor tweak. This discovery really overturns some established rules of mitochondrial genetics. Specifically, it challenges that whole threshold effect idea.
The idea that you need a lot of mutant mtDNA before you see a problem?
Exactly. These new mutations, they seem to be functionally dominant. That means even a tiny amount, a small percentage, can cause a massive change in the cell.
Okay, let's start with the evidence. To claim a whole new class of mutation, you need some serious data backing it up. How big was this study?
Oh, it was immense. They analyzed over 14,106 whole tumor genomes. And crucially, they got really deep sequencing coverage for the mitochondrial DNA.
So they could be sure they weren't missing things, even low-level mutations?
Precisely. That let them spot these low-frequency variants accurately. And what immediately jumped out was that these recurring somatic mutations, these hotspots, they clustered right in the genes that build the mitochondrial ribosome.
Which are?
mtRNR1 and mtRNR2. They code for the small and large RNA subunits of the Mitoribosome.
And were these just occasional finds, or?
Not at all. These RNA genes were actually among the top-ranked genes for somatic hotspots across the entire mitochondrial genome. Only mtND5 had more.
Wow.
Yeah. They estimated that something like 4% of all the tumors they look at had at least one of these RNA hotspot mutations. That's a significant number.
Okay, but here's what's confusing me. Ribosomal RNA genes, they build the machinery that makes proteins. They're usually super conserved, right? Like, evolution doesn't mess with them much because changes are usually disastrous.
That's exactly the paradox. That's what makes this so wild. These RNA genes are normally under very strong purifying selection in the germline.
Okay, so for our listener, purifying selection basically means?
It means nature heavily weeds out any variations there. In healthy people, mutations in these spots are almost never tolerated because they'd likely cause severe disease or just wouldn't be compatible with life. You can check databases of healthy mitochondrial DNA like Helix mtDB, and you just don't see functional mutations in mtRNR1 and RNR2.
Except for maybe a couple of rare ones linked to hearing loss with antibiotics, but not major mitochondrial diseases.
Right, exactly. But here, in cancer, you have cells actively selecting for mutations in these highly constrained, normally off-limits positions. It tells you something really fundamental has changed in the cancer cell's biology. It's like it has permission to break rules healthy cells can't.
Okay, that setup perfectly leads into the main breakthrough here. Because normally with mtDNA mutations, we talk about heteroplasmy, right? The percentage of mutant copies versus normal ones.
Right. And the standard rule for pretty much all known hereditary mitochondrial diseases and even the mtDNA mutations we previously knew about in cancer is that they're functionally recessive.
Meaning you need that threshold effect.
Yeah. You typically need a high percentage of mutated mtDNA, often over 50 or 60%, sometimes even higher, before the cell's energy production really tanks and you see a disease phenotype. You have to break, a lot of the power plants before the lights start to flicker.
Okay, but this research flips that script completely. You said these mtRNA hotspots are functionally dominant. How do they actually prove that just a little bit of this mutation causes big problems?
So they zeroed in on one specific hotspot, M.1227GA, as their main example, and they used this really precise tool, mtDNA base editing, to introduce it into cells at controlled amounts.
So they could dial up the percentage of the mutation.
Exactly. And this specific mutation, M.1227GA, it's in a really critical spot structurally. It's in the head domain of the small ribosomal subunit, the MTSSU, and it messes with how the A site tRNA binds. Basically, it gums up a key part of the protein production line in the mitochondria.
So it's not just slowing things down, it's like causing a jam.
That's a good way to put it. A jam in the works.
Yeah.
And the functional studies confirmed that. They did multimodal profiling and saw this really clear, specific loss of key respiratory chain proteins.
Like which ones?
Things like the complex 1 subunit NDU-FB8 and the complex 4 subunit MTCO2, proteins essential for energy production.
Okay, and here's the kicker. At what percentage of mutation, what heteroplasmy level did they see this protein loss?
This is the crucial part. They saw this significant functional loss when the mutation was present at only about 10% to 40% heteroplasmy.
Wow, only 10 to 40%. That's way below the usual 60% threshold.
Way below. It's shockingly low. And that's what confirms the functionally dominant nature. Even a small fraction of faulty ribosomes is enough to drag down the whole system and cause a major problem with cellular respiration. It basically breaks that old threshold rule. Okay, so we've got this functionally dominant mutation jamming up the Mitoribosome at low levels. What's the immediate fallout for the cell's energy production?
Well, I guess a big hit to the main energy pathway, oxidative phosphorylation, OXPHOS.
Exactly right. They measured the basal oxygen consumption rate, the OCR, and saw a substantial drop. The thinking is that the mutation causes the Mitoribosomes to stall.
Like a traffic jam on the assembly line.
Yeah, a permanent traffic jam.
Yeah.
And that stops the formation of functional monosomes.
Monosomes being the complete working mitochondrial ribosome.
Correct. If you can't form working monosomes because they're stalled, you can't properly translate the proteins encoded by the mtDNA, like those complex 1 and complex 4 subunits we mentioned. And this happens even if the cell still has a normal amount of mitochondrial DNA overall.
Okay, so the main power system, OXPHOS, is failing. But cancer cells are notorious for adapting. What did these cells do to compensate?
They switch gears metabolically. They start guzzling nutrients. The cells with the M1227GA mutation cranked up their intake of glucose, glutamine, pyruvate, basically any fuel they could get.
And shift towards glycolysis, burning sugar faster but less efficiently.
Precisely. They saw all the signs of enhanced glycolysis, higher levels of intermediates like glucose 6-phosphate, and also metabolites tied to the cell's redox state like lactate and malate.
So that compensatory rush must really change the cell's internal chemistry.
It definitely does. It alters the crucial NAD plus to NADH ratio, which in turn drives that ramped up glycolytic flux. It's basically trying to make up for the failing OXPHOS by running the backup generator flat out. And interestingly, this metabolic state mimics what we used to think required those high heteroplasmy recessive mutations, but here it's happening at maybe 1/5 the dose.
But it gets even more interesting when they look deeper into the TCA cycle, that central hub for both energy and building blocks. They use labeled fuels, right? Isotopes?
Yes, isotopically labeled glucose and glutamine. This lets you track not just how much of something is there, but how fast the cell is processing it, the metabolic flux.
What did that show? Was the cycle slowing down because OXPHOS was weak?
Counterintuitively, no. They found this sort of metabolic hyperplasticity. While the total amount, the pool size, of some TCA metabolites like citrate and aspartate actually went down.
Meaning the cell was running low on those specific parts?
Kind of, yeah, but the flux, the rate at which new material was flowing through the TCA cycle, was actually greater in the mutant cells.
Huh. So the engine's inefficient and low on some reserves, but it's pushing fuel through faster than ever.
That's what it looks like, including what's called anaplerotic labeling, how quickly they're stuffing new material into the cycle from the sides. This suggests an enhanced capacity for both breaking down fuel, catabolism, and building new stuff, anabolism.
Which cancer cells need for growth.
Exactly. That rapid turnover, supported by glycolysis, provides the building blocks, nucleotides, amino acids needed for aggressive proliferation. So this metabolic rewiring, driven by a low-dose mutation, really looks like a key advantage for the tumor. So let's pull this all together. What we're looking at seems to be a genuinely new class of cancer driver mutations, these mtRNA hotspots. They are, as far as we know, the first recurrent, functionally dominant mtDNA mutations described in any human disease.
And they cause this dramatic remodeling of metabolism and protein production at really low levels, rewriting the textbook on mitochondrial genetics.
Pretty much. They operate well below that classic threshold, showing that rules we thought were fixed might not apply in the context of cancer.
It's still amazing the cell can even tolerate these mutations in such a critical piece of machinery. And you mentioned something fascinating earlier, that the problems in the mitochondrial ribosomes seem to also affect the cytosolic ribosomes, the ones out in the main cell.
Yeah, that was an intriguing observation in the data. It suggests there might be some kind of coordinated regulation or stress signaling happening between the mitochondria and the rest of the cell's protein-making machinery.
So it's not just a local problem in the mitochondria, it's sending ripples throughout the cell.
It could be. It hints that the mitochondrial state has a really potent influence, and it reinforces this idea that cancer operates under different rules. The researchers called it the biologically permissive lens of cancer.
Meaning cancer can get away with mutations that would normally be lethal.
Essentially, yes, it can select for these, well, exotic genetic changes in fundamental machinery, like RNA, because the usual constraints that apply to healthy cells are somehow bypassed or maybe even exploited.
Yeah.
And maybe that brings us to a final thought to leave you with. Normally, mutating essential, non-redundant parts of the central dogma, like ribosomal RNA, is just incompatible with life.
Right.
So the fact that cancer cells are actively selecting for and seemingly thriving with these functionally dominant RNA mutations, what does that really tell us about the absolute rock-bottom requirements for a cancer cell to survive compared to the much stricter requirements for a healthy cell? It really makes you think about what's truly essential under different biological pressures.
A really fascinating question. What rules can be bent or broken when survival is the only goal? These tiny mitochondrial mutations certainly seem to be driving some very big consequences in how tumors behave. Well, that's all the time we have for this deep dive. Thanks so much for joining us. Remember to like and subscribe. It really supports the science experience, and we'll catch you next time for another exploration into the latest science.